In today's class, we are going to discuss the new chapter, photosynthesis. So, all of you know about photosynthesis, that it is a process by which green plants would make their food. That's the simplest way how you can explain the process. But in this chapter, we'll be discussing the process of photosynthesis in details. So you will learn that photosynthesis is a highly complex biochemical process which occurs in the green parts of the plant, and in the presence of sunlight and chlorophyll, finally carbon dioxide would be converted into sugar, where oxygen will be liberated as a byproduct. Photosynthesis, the entire reaction can be summarized in this equation, that is carbon dioxide, water in the presence of sunlight and chlorophyll is converted into sugar, and oxygen will be released as a byproduct. This is the equation in which we can summarize the whole process of photosynthesis. Alright. Now, during photosynthesis, the plants are getting to make their food, fine, but then do remember, photosynthesis is the primary source of food for all living organisms. We all living organisms are directly or indirectly dependent on photosynthesis for our energy source. Also, the major advantage of photosynthesis is that it would release oxygen as a byproduct, which will fill our atmosphere with oxygen, and hence it will allow the aerobic life forms to evolve. Okay? So now let us start with a few important experimental demonstration on the process of photosynthesis. Alright? Now, suppose we know that for photosynthesis to occur, certain of the things are very much necessary, like sunlight, chlorophyll, carbon dioxide. These are mandatorily required for a plant to undergo photosynthesis. Now, let us discuss a simple experiment by which we can prove that sunlight and chlorophyll both are required for photosynthesis to occur. Okay? So, experiment to demonstrate sunlight and chlorophyll are necessary for photosynthesis. Now see how we will do so. Simply for this demonstration or for this experiment, you need to take a de-starched potted plant. Okay? A de-starched potted plant means a plant which has been kept for a considerable period in a dark room where there was no light available. Hence, no photosynthesis would occur and all the stored starch will be used up. Hence, the plant will get de-starched. Okay, so we need to take a potted plant which is de-starched, also which should possess variegated leaves. Means leaves which have patches of green or white, so that the green patches would be filled up with chlorophyll. Alright, so we take a potted plant and which is de-starched, okay, and before we expose it to sunlight, we would finally wrap a few of its leaves with black paper so that those leaves could not be or will not be able to access sunlight. Alright, so we will take this de-starched potted plant with variegated leaves and then before we expose it to sunlight, we will wrap up a few leaves with black paper and then we will finally keep it in sun. We will allow it to photosynthesize for a few hours, and after that we will take a normal leaf and a leaf which was wrapped up. Say this is the normal leaf and a leaf that was wrapped or covered. Okay, so when we will allow both the leaf to undergo starch test, that means by putting a few drops of iodine solution, you know very well, if iodine comes in contact with starch, it will undergo color change. Bluish black color will be produced. So we will see that the leaf that was covered would not be producing or would not be giving a positive starch test. The positive starch test will be only produced by the normal leaf which was not wrapped up. Also, in this leaf, only those green patches, okay, because it's a variegated leaf, so those green patches which have chlorophyll, they would be undergoing positive starch test. By this, it can be easily proven that sunlight and chlorophyll both are required for photosynthesis to occur. Alright, understood this? Fine. Now, next, let us discuss a few important discoveries, early discoveries associated with photosynthesis. So, related to early discoveries, first is Joseph Priestley. Joseph Priestley in 1770, he conducted the famous bell jar experiment. Okay. So in the bell jar experiment, inside a bell jar, he would first introduce a burning candle and he would eventually see that the flame will be extinguished. It will be put off. Okay. Of course, today we know that is because of the cut in the, or rather in the, it will be happening due to the diminished level of oxygen, isn't it? But then at that time in 1770, oxygen was not discovered at all. Oxygen got discovered in 1774. It is because of this reason that at that time this simple discovery was regarded very important. Alright. In the next bell jar, a mouse was introduced. A live mouse was introduced. Of course, due to lack of oxygen, eventually we'll find that the mouse would die, right? But in the third bell jar, when Joseph Priestley introduced a burning candle and a mouse, plus this time he would also introduce a small mint plant. Okay, this time inside the bell jar he will find that neither the candle would be put off nor would the mouse die. So from this he would draw a conclusion that maybe something in the air was being damaged by the burning candle and the mouse, which was getting replenished by the plant. Okay, so green plants would replenish what was being damaged by burning candle and mouse. Alright, understood this? Now, the next important discovery was made by Jan Ingenhousz. He further reconfirmed what was being suggested by Joseph Priestley. Okay, so he extended kind of whatever was said by Joseph Priestley, he simply extended that particular thing. So what he did is that Jan Ingenhousz, he experimented with the aquatic plant Hydrilla. He observed that during the daytime in the water nearby to the Hydrilla plant, certain bubbles will arise, and he proved these bubbles to be oxygen. But he found that those bubbles will be missing or will be absent at night. So from this he would conclude that plants will undergo a process by which oxygen will be liberated, but then that process will only occur in the daytime, not at night. Okay, so it is an extension of what Joseph Priestley had suggested. Next, Julius von Sachs. Julius von Sachs, he is a German botanist, and he basically have given a various contribution in the field of botany. So Julius von Sachs, what he suggested regarding photosynthesis is that in the photosynthesis process, the product is going to be glucose, which is stored in the form of starch, which is a very important information that was proposed by Julius von Sachs. Not only that, he also suggested that there are certain green color bodies present inside the plant which contains green color pigments, due to which photosynthesis occurs. At that time, the term chloroplast or chlorophyll were not coined yet. Okay, so these are very important things. He suggested the photosynthetic product is glucose, which is stored as starch. Alright, next, T.W. Engelmann. T.W. Engelmann, he proposed the first action spectrum of photosynthesis. Okay, that means basically in T.W. Engelmann's experiment, he experimented using a green algae Cladophora and a suspension of aerobic bacteria. We know very well that photosynthesis occurs in the visible spectrum of light, isn't it? But then, visible light consists of 7 different colors. In which color does photosynthesis occurs in the most efficient manner? That was found out by T.W. Engelmann by his experiment with Cladophora and a suspension of aerobic bacteria. So what he did is this. Suppose this is the slide. Okay, in the compound microscope, you know, there is a stage present in which we keep a slide, and below that remains a mirror by which we direct the light to illuminate the stage, isn't it? So suppose this is the slide in which Cladophora was kept, and then on the surface the aerobic bacterial suspension was spread. Below the stage remains the mirror. Suppose this is the mirror. But T.W. Engelmann, he designed it in a manner that in between the stage and the mirror, he kept a prism. You know very well that the light will be splitted into its 7 colors by the prism. Okay, so that means when the slide was illuminated, different parts of Cladophora would receive different colors of light. Right? He kept the apparatus like this for a few hours, and after that he observed that if there was any change. Okay, so what he observed was that by the time after a few hours, when the Cladophora had photosynthesized, most of the bacteria had gathered in 2 zones actually. A zone where blue light would fall, a zone where red light was there. Blue and red light. In those 2 zones, all of this aerobic bacteria would be gathering. From this, he would conclude that because byproduct of photosynthesis is oxygen, isn't it? So in that case, aerobic bacteria would on the other hand require oxygen for their metabolism. So that's why in the zone where blue and red light were falling, there the photosynthesis was occurring most efficiently. Hence, their oxygen was also being released at a much higher rate, and that's why all the aerobic bacteria would gather in the blue and the red zone. Okay, so from this experiment it was proven that yes, photosynthesis does occur in visible spectrum of light, but then it most efficiently would occur in red and blue radiation only. Okay, understood this? Next, Cornelius Van Niel. Cornelius Van Niel, he experimented with purple and green sulfur bacteria. Purple and green sulfur bacteria, what he did is that he used this bacteria to finally resolve a long undergoing debate that when photosynthesis occurs, a group of scientists, they believe that the oxygen that is released during photosynthesis, it arises from carbon dioxide. But Cornelius Van Niel, by his experiments with purple and green sulfur bacteria, he proved that no, during photosynthesis, the oxygen that is released, it will arise from water, not from carbon dioxide. So that was a very important discovery that resolved all the debates related to this topic. Alright, so these are important early discoveries related to photosynthesis. Now finally we can start discussing the process. Alright. Site of photosynthesis. Where exactly in the plant body photosynthesis occurs? Okay. Now see, photosynthesis occurs in the chloroplast. The main site where photosynthesis occurs is the chloroplast. Fine. So chloroplast, if you remember the structure of chloroplast, it is a double membrane bound cell organelle, a green plastid, consisting of mainly two regions or two parts. A simple diagram I am making for you to understand in a clear manner. So this is suppose the structure of chloroplast having a double membrane. It mainly consists of two parts. This is called the grana, that is formed by piles of thylakoids stacked upon one another. These are the interconnections between the grana called stromal lamellae. We can also call them frets. And finally is the matrix of the chloroplast, which is called the stroma. So we can say that mostly the chloroplast consists of two parts, grana and stroma, isn't it? Now see, photosynthesis being a highly complex biochemical process, we can divide the entire mechanism of photosynthesis into two phases. First is called the light reaction, second is called the dark reaction. Light reaction is the one in which sunlight is entrapped and converted into chemical energy in the form of ATP and NADPH. Dark reaction is the second phase, and in the second phase, basically using the ATP and NADPH which is already formed, carbon dioxide would be converted into sugar. Alright, understood this? So light reaction is for making energy molecules, and dark reaction is to finally use those energy molecules to form sugar. Understood this? So in the chloroplast, there is a clear-cut division of labor that can be seen, because the light reaction occurs in the grana and of course stromal lamellae, and the dark reaction would occur in the stroma. Okay? Light reaction in the grana, dark reaction in the stroma. So we can say that chloroplast shows a clear-cut division of labor. Okay? Now, let us observe the grana in details. Suppose this is a grana consisting of 4 thylakoids. 1, 2, 3 and 4. And these are stromal lamellae. Alright. Now, if you observe the grana, the membrane, the thylakoid membranes, you will find that the thylakoid membrane consists of various types of complexes embedded onto it. There are mainly 4 complexes present: PS1, PS2, cytochrome B6F complex and ATP synthase. Okay, these are the 4 types of complexes which majorly would remain associated with the thylakoid membrane. Now, if you look at the grana properly, you will find that there are 2 types of thylakoids here. 1 and 4. These are called non-oppressed thylakoids. See, they have free ends, not being pressed from both ends. So we call them non-oppressed thylakoids. 2 and 3 can be said as oppressed thylakoids. Right? Now see, PS1 is going to be mainly present in non-oppressed thylakoids and stromal lamellae. Alright? PS2 on the other hand will be mostly found in oppressed thylakoids. Alright? It will not be found in non-oppressed. Cytochrome B6F complex and ATP synthesis, they would be found equally distributed in both non-oppressed and oppressed thylakoids. So do remember this particular fact, that PS1 and PS2, they will be differentially distributed in the entire grana. It will not be present in all types of thylakoids, but cytochrome B6F complex and ATP synthesis will be found equally distributed in non-oppressed and oppressed thylakoids. What is PS1, PS2? I'll let you know later on. Okay. After this, let us discuss a little bit regarding photosynthetic pigments. So regarding photosynthetic pigments, these are the pigments which will entrap the sunlight and allow the formation of ATP and NADPH during light reaction. Now, photosynthetic pigments mainly are of 2 types, that is chlorophyll and carotenoids. First, talking about chlorophyll. Chlorophyll are the most abundant pigments that are present in the plants, which will allow photosynthesis or light reaction in photosynthesis to occur. Chlorophyll, chlorophyll pigments, they can be of 5 types reported in the plant body, that is chlorophyll A, B, C, D and E. Out of these, chlorophyll A and B are majorly found in higher plants. Okay, so because we are discussing here about photosynthesis in higher plants mainly, so we are concerned here with chlorophyll A and chlorophyll B. Now, if you ask me that out of chlorophyll A and B, which is the most important one? Of course, chlorophyll A is going to be in majority found in the plant body in comparison to chlorophyll B. Chlorophyll B will be approximately 1/4 of chlorophyll A found in the plant body. Okay, so chlorophyll A will be found in much more abundance in comparison to chlorophyll B. Now, there are certain important facts you should remember about chlorophyll A. Chlorophyll A is termed as universal photosynthetic pigment. Why is that so? Because chlorophyll A is found in all types of plants, even in cyanobacteria. So chlorophyll A is found in all types of photosynthesizing organisms except for a few bacteria maybe. That's why we call it universal photosynthetic pigment. Also, chlorophyll A is also termed as the primary photosynthetic pigment. Because you see chlorophyll A, chlorophyll B, carotenoids, all the types of photosynthetic pigments, they will try to entrap sunlight during light reaction. But the pigment that will actually participate in the light reaction is chlorophyll A. Rest of the pigments would not directly participate in the process. Okay, they will just simply be entrapping sunlight and then transferring it to chlorophyll A so that it can perform light reaction. Hence, we call chlorophyll A to be primary photosynthetic pigment, and rest of all the pigments, that is chlorophyll B and carotenoids, they are known as accessory pigments. Okay, understood that? Now, let me give you a little idea regarding the structure of chlorophyll molecule. Now, regarding chlorophyll, the structure is as such. This is in general the structure of chlorophyll pigment. Chlorophyll is a tadpole-like structure. That means a structure that has a head followed by a tail. Now, the head is known as porphyrin head, whereas the tail is known as phytol tail. Alright, the porphyrin head is made up of 4 pyrrole rings. So we call it tetrapyrrole ring. Okay, so this pyrrole rings, if you see, it is made up of 4 carbon atoms and a nitrogen that is projected towards the inside. So from the 4 pyrrole rings, this 4 nitrogen will hold a non-ionic magnesium at the core. Alright, and the pyrrole rings will be joined or attached to one another by the help of methane bridges. Fine. Now, the 4th pyrrole ring will finally be holding the tail, the phytol tail. The phytol tail is made up of 20 carbon alcohol unit with the formula C20H39OH. With the help of the phytol tail itself, the chlorophyll pigment will remain bound to the thylakoid membrane. Okay, the phytol tail will allow the pigment to be embedded into the thylakoid membrane. Fine, understood this? Now, without this magnesium, the chlorophyll will become colorless and will be termed as pheophytin, which is a early electron acceptor during the light reaction. Fine. Now, this being the main, all the chlorophyll that we know about, all of them will have basically a structure like this, a porphyrin head followed by a phytol tail. But then we have so many different forms of chlorophyll, chlorophyll A, B, C, D, isn't it? So what brings the difference then? What is the major difference? The major differences are brought about by the side chains. See, between chlorophyll A and B, the major difference in their structure lies in the 3rd carbon. In the 3rd carbon of chlorophyll, chlorophyll A, there will be a methyl group present. That means in the second pyrrole ring, there will be a methyl group present for chlorophyll A, which is going to be replaced with the aldehyde group in chlorophyll B. That's the major difference between the structure of chlorophyll A and B. Alright, understood this? Now, let us differentiate between chlorophyll A and B distinctly so that you can understand their basic difference in the nature. Fine? So, chlorophyll A versus chlorophyll B. Chlorophyll A versus chlorophyll B. Chlorophyll A is bluish green in color, whereas chlorophyll B is yellowish green, or we can say olive green in color. Chlorophyll A has the formula C55 872 O5 N4 Mg, whereas for chlorophyll B it is C55 870 O6 N4 Mg. Alright. Now, next, chlorophyll A and chlorophyll B. Do remember a very important fact here. Chlorophyll A or chlorophyll B, or the major photosynthetic pigments, chlorophyll and carotenoids. All of these pigments are water insoluble pigments. They will solubilize in certain other organic solvents but not in water. Alright. So in that case, chlorophyll A would readily dissolve in petroleum ether, whereas chlorophyll B will dissolve with 92% methyl alcohol. The most important thing that will be required later on for you to understand the light reaction as well, chlorophyll A mostly will absorb. See, you know very well now that photosynthesis occurs in blue light and red light, isn't it? Chlorophyll A will mostly absorb red light rather than blue light. It will absorb more of red than blue, whereas chlorophyll B will absorb more of B, then that means more of blue light than red. Understood this? So, it absorbs more of red light than blue, whereas it will absorb more of blue light than red. Okay, these are the major differences between chlorophyll A and chlorophyll B.